My name is Jong. I work at the Netherlands Academy of Sciences. We are one of the proud sponsors of this conference, and I'm enjoying it very much so far. My talk will be somewhat a repetition of what has been said before, but I think that's not necessarily a bad thing, because I will argue that repetition is very important in science. So here we go. Yes. The Royal Academy of Sciences advises on the proper conduct of science, and was informed that there were many concerns about the lack of reproducibility of study results, and wanted to investigate that issue, and especially whether replication studies as a means to solve this problem, if there even is a problem, should be stimulated, and how to do so. We worked with a committee, and we based ourselves on the scientific literature, reports by other advisory bodies, interviews with experts, and an invitational conference. And what I present here are our preliminary findings. The report will be due in October, after the summer. These are the people on the committee. The chair is Johan Makkenbach, and I'm at the bottom because I'm the secretary, not a professor. The rest is almost everybody's a professor. And this is the committee. Most of the people on the committee are in the life sciences and medical field, but we also have a psychologist, Wagemakers, and what I will say is largely based on those broad fields, medical and psychology, but we think our lessons or conclusions apply much broader than that, so to most empirical sciences. Scientific progress requires that the results are reproducible. Well, are they? Difficult to say. There's no good data across science. There have been a few samples in different fields. For example, the gentleman there, Brian Nosek, did a study of 100 psychology studies, repeating them. He found that 36% of them could be reproduced. But on the other hand, Amgen found only 11% in doing a repetition of preclinical studies. Is this a problem? Well, difficult to say, but when the study results of only 11% are similar to those of the original studies, you can have some doubts about that. But even if it's 85%, it doesn't mean that those results are actually true or that they are valuable. So even if it's 85%, maybe that's even too low. I can't say. The gentleman there, Mr. Gonon, said, there isn't a crisis. 52% of the readership of Nature disagrees with him. They think there is a big crisis, and 38% thinks there is a slight crisis. Some definitions may be boring, but I think necessary for now. A replication study, as we presented, is a study that is designed to test the reproducibility of the results of a previous study, so a repetition of a previous study. And the method should be, quite similar, because otherwise a good assessment of whether results can be reproduced is not possible. And you could say, results are reproduced. You can do all sides of statistical analysis, but maybe a more pragmatic solution would be to say, if the results are such the same, that it won't raise many questions about the previous study. So there we go. A little word about this. Even if a study is reproduced, it doesn't say whether it's true. It will increase some confidence in previous findings, of course, when you can reproduce a study, but it doesn't make them true. And on the other way around, if you can't reproduce a study, it doesn't mean the previous study was false. So there could be also, the previous study could be right, the next study could be right. You have to delve into the details to find out what's true and what's not. The impact of non-reproducibility can have many things. We already touched on those things, polluting the drug pipeline, delaying scientific progress, wasting resources. So the impact can be significant. One of the audience asked, what are the causes of non-reproducibility? And I won't go into this in detail, but there are many, many causes. There are already in the literature, many people wrote about it, but there are many, many causes. What I want to say here is, well, some of these factors, like noise or human errors in studies, are really inherent in the scientific endeavor and can't be ruled out definitely. There will always be irreproducible results. But on the other hand, there are other factors like shopping around in the analysis or too small sample sizes, which are avoidable. And some of these can even be considered questionable research practices. Well, there are a lot of measures you can take to prevent irreproducible results. You can do all kind of things to improve the study methods, improve the study reporting, and improve the organization and culture of research, so the incentive system in which researchers work. But I won't go into this because the other solution is not prevent reproducibility, which I think is very, very important, but solve it afterwards by doing a replication study. Well, there are many kinds of different replication studies, so there's no gold standard here. You can make a decision who will execute the study. Will be the same researchers, different researchers, a combination of the two. What aspects of the study will you actually replicate? Do you replicate the whole study or only the analysis? Do you gather new samples? There are many choices to be made there. And thirdly, and very importantly, how precisely will you follow the original protocol? Some say you should do it very precise, but sometimes it is not possible or feasible. For example, if you work with patients, then by definition you can't do the exact same study twice. You will have different patients in the next study. So, well, then the difficult question is, well, what percentage of research efforts should go into replication studies? Well, difficult to say. In most fields, it's only a few percent. But as I already said at the mic, the desired rate of replication should depend on well, the degree of non-reproducibility or the likelihood that previous results are non-reproducible. And also whether it's an efficient use of research funds. And you also have to bear in mind the alternatives, doing an innovative study or preventing irreproducibility. While good replication studies require that there is good information about the previous study, and currently that is not the case. Studies are not reported in enough detail and not enough data are available often to do a proper replication study. Yes. You do have to know when and how to perform a replication study. Well, you first have to start out with assessing the need for replication, and then you have to really be very careful in choosing the right study design and interpreting the study in terms of reproducibility. And some researchers now lack the skills to do that properly. And finally, you need to have the proper incentives for a replication study. Researchers, well, often don't really like them. They can see it as an attack on colleagues. Funding agencies, they focus on innovative research, journals also, and the career evaluations too. So what are the recommendations of the Netherlands Academy of Science? Now, towards research, well, do actually those replication studies when appropriate. And also do meta-science, generate data on reproducibility and replication study as a topic. Funders, well, they should start out with assessing the need for replication in a field. That should be the first step, and after that, they should fund replication study when needed. And already the NWO initiative has been mentioned. That has been 3 million for funding the replication studies in the Netherlands. Well, journals have a big role to play here too. Currently, they well, they sometimes discourage even replication studies, but they should really encourage them by special issues or by including them as one of the formats for a journal, for an article submission. They should provide detailed guidelines for how to report methods. For example, the journal Cell has the STAR methods approach, and that gives a very detailed guidelines for how to report your study, so that another researcher can, if needed, repeat it. And finally, journals and both institutions, I think, should require the storage of study data and methods. Well, this is my final slide. Institutions have a role to play here too. They and funders should require the pre-registration, and then especially of the hypothesis testing studies, so really not the exploratory studies, but of the confirmatory studies. They should educate researchers better on how to design their studies and assess reproducibility, and they should credit replication efforts in the career evaluations. This was my talk, and I'm happy to answer any questions.
Thank you very much, Jean-Philippe. I know that you're a philosopher, and it was clear to me that you are a philosopher. Who wants to ask questions or put a comment? Please use the microphone or come over here. There you go.
I have, I have a couple, but I'll start with one now. Did your study look, so one of the things, I just completed a study on integrity. I work at the US National Academies. And so it wasn't primarily about reproducibility or application or anything, but we had to kind of deal with that in some way. And so one of the questions that I don't know whether your committee addressed at all or thought about was, what would be the acceptable level of irreproducibility in a field? And it might be different in different fields. It might be just inherently, for example, in biomedicine, if you have small sample sizes and things like that, it might be an acceptable rate of irreproducibility would be higher than in some other fields. And how would you determine what that was? Did your committee look at that question at all? Or?
Yes, we did discuss that, and well, true, it will depend on the field. A few aspects to take into account. What is the impact of irreproducible results? Does it directly harm individuals, patients? Do bridges come down if your study is not in order? And also the cost involved would be a big factor. And I think the question whether to do a replication study is not so much different how you approach that in terms of the value of information you get from that than doing an original study. Also in that case you have to really be careful and think, well, what does this information add to the information already available, and what would be the impact of a new finding here, or in case of a replication study, what would be the impact of knowing a presumed fact better than you did before.
Maurice Segers, Maastricht University. I could have asked this question actually to any of the others, but you're the last one of this session. When I hear the topic of replication studies, for me it sounds very much just like meta-analysis. And all the topics that you talk about, small sample size, publication bias, pre-registration need, and it sounds all like déjà vu. It's like a meta-analysis. And a meta-analysis, actually, we have the advantage of heterogeneity, because a study has been reproduced in another sample, and still you find finding, or you summarize, you find nothing anymore. So what is the real advantage of a replication study, especially because it's only one replication study, and in meta-analysis we'll use it then look at the sample of studies, and we just have one study more. How much does that add?
Yeah, I agree totally that there's a big connection between replication studies and meta-analysis. In meta-analysis, presumably would go a lot of replication studies, because they address the same question with the same methods and can be compared, and sometimes even on a individual for example, patient data level, you can pool the data. So actually, you would need a lot of replication studies to do a good meta-analysis. So there's a strong link between them, sure. But I think in a medical field, the idea of doing meta-analysis is very well accepted, and that's not so much the case in all other fields. So they would have to take two steps, do more of the same studies when needed, and pool them together in a kind of a meta-analysis or another approach to pool the data together and get firm conclusions.
Gerrit Zielhuis from Nijmegen. Did your committee or yourself had the idea to copy the best practice of the geneticists who already for 10, 15 years now have as a rule that they only publish their explorative analysis when they did the validation themselves. So it's not so much doing replication studies, but just the final phase of the primary study. Did you consider that to expand that for the whole biomedical or even broader field?
Yeah, we did think about that because I talked, well, I didn't mention it, but it was on the slide. We defined a replication study as a separate study in which you replicate a previous study. So that doesn't tell replication within a study, doing a control experiment and publish them together, which is also a very common practice also in laboratory work. We wouldn't call that a replication study, and the main reason for that is that there isn't for those kinds of internal applications, there are not the problems with not getting funding and not getting published in journals. When you pull different experiments together and you present them together, whether in an application or to a journal, then you won't face, well, that has been done before. So that would be the main difference. We draw the line there.
But if we join forces, the funders and the journals and the research community themselves, we don't need these replication studies anymore if this is just a regular last phase of a primary study.
Yeah, true. If you would pool different labs or populations together and do several experiments and then together publish them, well, then the problem is largely solved. Yeah. But we use the salami methods often in medical sciences, and that is published the least possible data in one paper. So.
Hi, Chris Harkering from Tilburg University, a statistician. I just would like to make first one comment about the publication bias and meta-analysis. Publication bias actually shifts the estimated effect, so it's very different. You need replication to get unbiased estimates. But I would like to ask you, I'm very happy to hear many of these statements, because if the National Academy makes them, they carry much more weight, so very happy. Although one statement seems kind of behind, because is there still the need to assess whether we need replications? Because we've been talking about this for almost 12 years throughout different fields, and there are many reasons to assume that most of the research findings are false, so maybe we should do the opposite and assess which findings do not need replications.
Well, I agree to you with you to some respect. In many fields there is now some data how big the problem is with regard to reproducibility. In some fields there is very little data, but I do think one of the things that is lacking data is you have to get behind the fact that there isn't a reproducible result. You have to investigate the causes, the factors behind it, and there is not much data on that. So, and I think that would be, in some fields there is, but not in all fields. And I think there would be some value in getting additional data on that.
Okay, we should definitely talk more, because meta-research has quite some evidence on it.
Okay.
There's room for a final question, a brief question with a brief answer, please.
All right. I wanted to ask you actually the same question as I think the first question in this symposium was asked to the first speaker, because I know you are experienced in researching clinical trials as well. So could you maybe comment on the need or the ethical, the balance between the ethical imperative of equipoise and the need for replication studies? So if there is not genuine uncertainty about whether a randomized, whether one treatment or the other will be effective, can you then still ethically do a new replication study?
Yeah, and actually that's a bit of a repetition of the question which you've been asked before, but I think, no, of course, if there's not uncertainty about whether a certain intervention, for example, is effective, no, you shouldn't do another study. But you see, for example, in how the EMA approaches drugs, they require several studies. And afterwards, they require also post-marketing surveillance. So, and that isn't really a replication study, but they are saying, well, you can never be too sure. So even if there's good data, still do some follow-up research after that.
Final question.
Just a small comment. So it has not been explicitly said, but in clinical trials, the authorities require 2 confirmatory trials. So, I mean, there's no question about ethical justification. It's what they require. That's a standard thing. And the other thing is, lack of reproducibility may be due to lack of external validity. And there may be a biological reason why there's no reproducibility. So we should always keep in mind the biology may be different in some conditions than in other ones. And then you need to check if that's the case, yes or no.
Yeah, I agree. Yeah.
Thank you very much, Jean-Philippe.